It's amazing, isn't it, how interconnected everything is in the human body.
Oh, absolutely.
We often think of our organs as these separate entities, like the heart does this, the lungs do that.
Right. Working independently.
Exactly. But what if, and this is a fascinating concept, what if a problem in one part of your body, like say your bladder, could actually make you more sensitive to pain in a completely different area, like your gut?
You're hitting on a really intriguing area of research, and actually, that's exactly what we're diving deep into today.
Perfect.
There's this emerging understanding of what's being called an interorgan neuroimmune circuit. And
Ooh, I like the sound of that.
yeah, it basically describes how inflammation in one of these internal organs, what we call visceral organs, can lead to a heightened sensitivity to pain in another one, even if that second organ doesn't show any signs of inflammation itself.
Okay, so we're talking about like the bladder and the colon, two
Mhm.
seemingly unrelated organs. But we know clinically that conditions like interstitial cystitis, ICBPS, and irritable bowel syndrome, IBS, often occur together, right?
You're absolutely right. The co-occurrence of ICBPS and IBS is a well-documented phenomenon. In fact, some estimates suggest that between 30 and 75% of people with ICBPS also experience symptoms that are very much like IBS.
That's a huge overlap.
It is. And it's something that has puzzled researchers and clinicians for a long time.
So the big question has always been why? Why are these two conditions so often linked? What were some of the prevailing theories before this new research came along?
Well, a lot of the thinking focused on these sort of global or systemic factors.
I like.
So things like, hormones
Yeah.
that circulate throughout the body. Perhaps they were somehow playing a role in amplifying pain signals.
Okay.
Or maybe there was some kind of miscommunication happening within the central nervous system, the brain and the spinal cord.
Right, the control center.
Exactly. The idea being that the brain might be misinterpreting pain signals from one area and kind of spreading that hypersensitivity to another.
But this new research, what we're diving into today, points to something much more direct, doesn't it?
It does. This research proposes a very exciting new hypothesis
Okay.
that there's actually a direct line of communication, a dedicated pathway, if you will, between these peripheral organs like the bladder and the colon,
Okay.
and that this communication is driven by these neuroimmune interactions, happening locally, right there in the periphery, not necessarily involving the brain or hormones in a major way.
This is really interesting stuff. So how did the researchers set out to explore this idea of bladder inflammation leading to problems in the gut? Did they poke a bladder and then watch the colon?
Well, not quite poking,
Okay.
but they did use a mouse model, very cleverly designed to mirror what we see in humans with ICBPS.
Okay.
So what they did was they introduced this antimicrobial peptide called LL37 into the bladders of these mice.
LL37, and why is that significant?
Well, LL37 has been found in elevated levels in the urine of people with ICBPS.
Oh, interesting.
So it's thought to be a trigger for the inflammation and pain that characterize that condition.
So by putting it into the mouse bladders, they were essentially inducing an ICBPS-like state.
Exactly. They were creating a model that allowed them to study those early events,
Wow.
the mechanisms that might be driving the connection between bladder inflammation and the gut issues.
And what did they observe? When they induced this bladder inflammation in the mice, what happened?
Well, first of all, in the bladder itself, as expected, they saw classic signs of ICBPS.
Okay.
So the mice started showing increased sensitivity to touch in the pelvic area. They call that allodynia,
Oh, okay.
which basically means that even light touch is perceived as painful. They also saw edema, swelling, in the bladder wall. And there was this infiltration of immune cells, these PMNs, or polymorphonuclear leukocytes.
Humans.
Yeah, those are the first responders, the hallmark of active inflammation.
Oh, okay.
And of course, the mice also exhibited urinary urgency, needing to pee more frequently. And very importantly, they became much more sensitive to their bladders being filled or distended.
Right. Makes sense.
The researchers actually measured this by looking at what are called visceromotor responses, or VMRs, which are basically reflex contractions of the abdominal muscles that happen when an organ is stretched.
So it's like a measurable physical response to that internal discomfort?
Exactly. A way to quantify the sensitivity.
So the bladder's clearly in distress. But what about the colon? Did it show the same kind of inflammatory response?
And this is where it gets really fascinating. Despite the fact that there was all this inflammation going on in the bladder, the researchers found no signs of inflammation whatsoever in the colons of these mice.
Really?
Yeah. And not only that, the lining of the gut, what we call the epithelial barrier, remained completely intact. No leaks, no changes in permeability.
So no direct inflammation in the colon, yet we know that these gut problems are often present in people with bladder issues. How did the researchers reconcile this? How did they know that the colon was actually being affected if it wasn't showing signs of inflammation?
Well, they looked very closely at the colon's function, both in terms of its sensory perception, so how it senses stimuli, and its motor activity, so how it moves things along.
Okay.
And what they found was that even though there was no inflammation, these mice with bladder inflammation developed a phenotype that was similar to constipation.
So like a slowing down of the gut's normal movements.
Exactly. They measured this by testing how long it took the mice to expel a small bead that they had put into the colon. And it took the mice with bladder issues significantly longer.
Oh, wow.
More importantly, they also found that these mice had an increased sensitivity to their colons being distended.
Meaning their colons were more easily irritated.
Exactly. And again, they measured this using VMRs, just like they did for the bladder.
So basically they were seeing a heightened sensitivity in the colon, even though there was no direct inflammation there. It's like the bladder's distress was somehow being transmitted to the colon.
That's a great way to put it. And this finding was really key because it provided strong evidence that the bladder inflammation could indeed trigger these IBS-like symptoms in the gut, even in the absence of any local pathology in the colon itself.
Okay, so the connection is becoming clear. Inflammation in one place leading to increased sensitivity in another. But how is this happening? How are these two organs, seemingly so distant from each other, communicating?
Well, that's the million-dollar question, right? Yeah. And this is where the concept of what are called polyorganic sensory neurons comes into play. So the researchers, based on their observations, they started to think, okay, maybe there's a direct neural circuit connecting the bladder and the colon. Maybe that's how they're talking to each other.
So a direct line, like a phone line, between these two organs?
Yeah, that's a good analogy.
And how did they go about mapping these neurons? How do you even begin to see these microscopic connections?
They used a really elegant technique called retrograde tracing.
Retrograde tracing.
Yeah. So they injected these fluorescent dyes, actually two different colors of cholera toxin B or CTB, into the bladder and the colon of the mice.
Okay. Two different colors, one for each organ.
Exactly. So these dyes, they get taken up by the nerve endings in the organs, and then they travel backwards up the nerve fibers all the way to the cell bodies of those neurons, which are located in clusters called dorsal root ganglia, or DRGs.
DRGs, got it. And these ganglia are located near the spinal cord.
That's right. They're kind of like relay stations for sensory information coming from the body.
So essentially, if a single neuron had connections to both the bladder and the colon, it would light up with both colors.
You got it. And that's exactly what they saw.
Really?
Yep. While most of the neurons were specific to either the bladder or the colon, they found that a significant portion, about 15.5% of the neurons they looked at, showed both colors.
Wow, that's a pretty decent chunk of neurons.
It is, and it really pointed to this shared neural pathway. Like these neurons are acting as these dual sensors picking up information from both the bladder and the colon.
Fascinating. And this wasn't the case with the autonomic nervous system.
Right, so the bladder and the colon, they also receive input from the autonomic nervous system, which controls things like, involuntary muscle contractions and heart rate.
Okay.
But when the researchers looked at the autonomic neurons that connected to these organs, they found very little overlap.
So the autonomic nervous system wasn't really the key player in this interorgan communication. It was those shared sensory neurons.
Exactly. They seem to be the main conduit.
But, okay, just because these neurons are connected to both organs doesn't necessarily mean that they're actually firing in response to signals from both, right?
Mm-hmm.
How did the researchers test that?
That's a great point. Anatomy is one thing, but function is another.
Right.
So they did these really cool experiments using in vivo calcium imaging.
In vivo calcium imaging. Sounds complicated.
It's a technique that allows them to visualize the activity of neurons in real time in living animals. So they used mice that had this special fluorescent calcium indicator gets into their sensory neurons. And then they separately distended or stretched the bladder and the colon in these mice. And they watched to see which neurons lit up, because when a neuron fires, it releases calcium, which then binds to this indicator and causes it to fluoresce.
So they could literally see which neurons were being activated by the bladder and which ones were being activated by the colon.
Precisely. And what they found was that about 7.8% of the sensory neurons that responded to bladder distension, they also responded to colon distension.
So those neurons were firing in response to both. That's pretty definitive proof that they're functionally connected.
Yeah, and that's how they came up with the term polyorganic sensory neurons, to describe these neurons that can respond to stimuli from multiple organs.
So we've got these shared sensory neurons acting like a direct communication pathway between the bladder and the colon. And it's through this pathway that the bladder's inflammation seems to be spreading that hypersensitivity to the colon. So my question is, could they actually block this transmission? Could they somehow silence those specific neurons and see if that would prevent the colon from becoming hypersensitive?
That's exactly the next step they took.
Of course it is.
Science. They used a combination of really sophisticated techniques called intersectional genetics and chemogenetic inhibition, using something called DREDDIes,
Dreads?
designer receptors exclusively activated by designer drugs. It's a mouthful, I know.
Okay, well, let's stick with DREDDs. What did they do with these DREDDs?
Well, basically, they used viruses to deliver these genetic instructions into the mice. And these instructions would cause those polyorganic sensory neurons, and only those neurons, to express this special inhibitory receptor.
So they were targeting those specific neurons that connected the bladder and the colon.
Precisely. And then they could activate that inhibitory receptor with a specific drug, so they could essentially silence those neurons on demand.
Like flipping a switch. Turn those neurons off and see what happens. And what did happen? When they silenced those polyorganic neurons in mice that had bladder inflammation, what did they observe?
The results were very interesting. So silencing those neurons, it didn't actually affect the bladder hypersensitivity itself. Those mice still had the bladder pain and the urinary urgency.
Oh, interesting. So the inflammation in the bladder was still driving those symptoms, regardless of what was happening with those shared neurons.
Right. But here's the key finding. Silencing those neurons did significantly reduce the colonic hypersensitivity and the constipation-like dysmotility that they had seen in these mice.
Wow, so they were able to essentially prevent the bladder's inflammation from spreading to the colon by shutting down that communication pathway.
Exactly. And that provided really strong evidence that these polyorganic sensory neurons are indeed the key players in transmitting that irritable state from the inflamed bladder to the colon.
This is really remarkable. It's like finding a specific wire that's carrying a signal and then cutting it.
That's a great way to put it. And this was a major advance in our understanding of how these two organs can influence each other.
So we've got these shared sensory neurons that act like a communication pathway, but what about the role of mast cells? I know they're often involved in inflammatory responses.
Yes, mast cells are really critical players in this whole process as well. They're these immune cells that are found near barrier surfaces in the body, like the lining of the bladder,
Okay.
and they're known to interact with sensory neurons, often contributing to pain and inflammation.
And where did they find these mast cells in relation to the sensory nerves in the bladder? Were they just kind of hanging out nearby, or was there a more intimate connection?
Well, they use some really cool imaging techniques, including whole-mount tissue clearing and 3D rendering, to visualize the bladder tissue in great detail. And what they saw was that while some mast cells were in the muscle layer of the bladder, the majority of them in the lamina propria, which is this connective tissue layer just beneath the bladder lining, were in very close proximity to, and in some cases even directly contacting, these nociceptors.
Nociceptors.
Those are sensory neurons that are responsible for detecting painful stimuli.
Oh, okay.
And specifically, they were looking at nociceptors that express this protein called TRPV1, which is involved in sensing heat and pain.
So the mast cells were getting up close and personal with the pain-sensing neurons in the bladder. And was this particularly evident in the areas where they had seen the inflammation after introducing the LL37?
Yes, absolutely. That close physical relationship really suggested that the mast cells might be involved in sending signals to those neurons, contributing to that increased sensitivity.
So how did they figure out if the mast cells were actually contributing to the interorgan sensitization?
They used a special type of mouse called a mast track mouse.
Mast trick. Okay, another one for the glossary.
In these mice, you can actually selectively deplete mast cells by giving them diphtheria toxin.
Wow, that sounds a little intense.
It is a powerful technique, but it allowed them to see what would happen in mice with bladder inflammation if you took the mast cells out of the equation.
And what did happen?
Well, not surprisingly, when they depleted mast cells in the mice with LL37-induced bladder inflammation, they saw a decrease in the bladder inflammation and hypersensitivity, which makes sense because mast cells are well-known players in inflammatory responses.
Okay, yeah.
But here's the really interesting part. Depleting the mast cells also significantly reduced the colonic irritability and dysmotility that they had seen in these mice.
Wow, so the mast cells were not only contributing to the inflammation in the bladder itself, but they were also necessary for that inflammation to spread to the colon.
That's right. They were clearly playing a key role in this whole interorgan communication process.
So it seems like the inflammation in the bladder triggers these mast cells, and the mast cells somehow talk to those polyorganic sensory neurons, and that's what leads to the colon becoming hypersensitive as well.
That's a good summary of the sequence of events.
But what kind of signals are the mast cells sending? What are the actual molecules involved in this crosstalk?
They look at several different pathways, but one that stood out was the involvement of a leukotriene C4, or LTC4, and its receptor, cis-LTR2.
Okay.
LTC4 is this lipid mediator that's released by mast cells, and it can bind to cis-LTR2, which is found on sensory neurons.
Okay.
And what they found was that if you block that receptor, either with a drug or by deleting the gene for it specifically in the bladder sensory neurons, you also get a reduction in both bladder and colon hypersensitivity.
So blocking that communication pathway at the level of the sensory neuron also seems to disrupt the spread of the hypersensitivity. Fascinating. And there's also this other receptor, Mr. PRB2, that's involved, right? And this one is found on the mast cells themselves.
You're right. Mr. PRB2 is this key sensor on mouse mast cells that can be activated by a variety of stimuli.
Okay.
And what the researchers found was that the vast majority, like over 96%, of the mast cells in the bladder express Mr. PRB2. And they're particularly enriched in that lamina propria layer, where we saw those close interactions with the nociceptors.
So the thinking is that this MSPRPRB2 receptor is what initially senses the problem in the bladder.
Exactly. It's like the alarm bell that sets off the whole cascade.
And to test this, they looked at mice that didn't have the MSPRPRB2 receptor.
Precisely. They used these MSPRB2 deficient mice, and they found that in these mice, when they induced that ICBPS-like condition, the bladder inflammation, the urinary urgency, and the bladder hypersensitivity were all significantly reduced.
That makes sense. If you take away the alarm bell, the mast cells are less likely to get activated.
Exactly. But here's the crucial finding. They also saw a substantial reduction in the comorbid colon irritability and dysmotility in these mice.
So even though the bladder was still being challenged, without that MSPRB2 receptor on the mast cells, the hypersensitivity wasn't spreading to the colon. That's a pretty big deal.
It is. It really points to MSPRB2 as a key initiator of this whole interorgan communication pathway, like it's the first domino to fall.
Okay, so in mice, the story seems pretty clear. Inflammation in the bladder activates mast cells through this MSPRB2 receptor. The mast cells then release signals that act on these polyorganic sensory neurons, and ultimately that leads to increased sensitivity in the colon. But what about humans? Does any of this translate to our biology?
That's the key question, right? And this is where the human counterpart of MRGPRB2 comes in. It's called MRGPRX2.
MRGPRX2. Okay.
And when the researchers looked at data from the GTADEX portal, which is this huge database of human gene expression, they found that the urinary bladder actually has the second highest level of MRGPRX2 expression out of all the tissues they looked at.
Really? So it's highly expressed in the human bladder, just like MRGPRB2 is in mice. That's a pretty good sign that it might be important.
It is. And to further confirm this, they looked at single-cell RNA sequencing data from human bladder tissue.
Single-cell RNA sequencing, which allows them to look at gene expression in individual cells, right?
Exactly. And what they found was that MRGPRX2 is specifically expressed by mast cells in the human bladder.
Okay, so mirroring what they saw in mice.
Precisely. The same cell type, the same receptor. It's very compelling.
And to take this even further, they used these humanized mice.
You're right. They used mice where they had replaced the mouse MRGPRB2 gene with the human MRGPRX2 gene. So now they had a model where they could study the function of the human receptor in a living system.
That's really clever. And in these humanized mice, did they see the same mast cell-specific expression of MRGPRX2?
They did. It was very clearly expressed on the mast cells in the bladder. So they had successfully recreated that key aspect of human biology in the mouse model.
And then, I'm guessing, they tested whether blocking MRGPRX2 in these humanized mice would have the same effect as blocking MRGPRB2 in the regular mice.
Exactly. And they did this using a new, very potent MRGPRX2 antagonist called EP001.
EP001.
First, they did some in vitro studies. So they took mast cells from these humanized mice and exposed them to a compound that specifically activates MRGPRX2.
Okay.
And they found that EP001 could completely block that activation. So they knew that they had a drug that could effectively shut down this receptor.
So they had the target, they had the drug. Now the big question, what happened in the living mice? when they gave EP001 to the humanized mice that had been induced to develop those ICBPS-like symptoms.
Well, the results were very encouraging. Just like in the mice that lacked the MIGPRB2 receptor altogether, treatment with ET001 in these humanized mice led to a big decrease in bladder inflammation, urinary urgency, and bladder hypersensitivity.
Okay, so it was working on the bladder itself?
Yes, but importantly, it also reduced the colon irritability and dysmotility that was induced by the bladder problem.
That's fantastic. So even in this humanized model, blocking that MRGPRX2 receptor seemed to prevent the spread of hypersensitivity from the bladder to the colon. This has huge implications for how we understand and potentially treat these conditions in humans.
I agree. It's a very promising finding.
So this has been a mind-blowing journey through some really cutting-edge science. To wrap things up, what are the key takeaways from all of this? What's the big picture here?
I think the main takeaway is that we now have a much clearer understanding of how this direct communication pathway between the bladder and the colon works. We've identified these specialized neurons, the polyorganic sensory neurons, that connect these two organs.
Right. And we've also seen how crucial mast cells are in initiating this communication, specifically through that MIGPRB2 MRGPRX2 receptor.
Absolutely. That receptor seems to be a key player in sensing the initial problem in the bladder and setting off the cascade of events that ultimately lead to hypersensitivity in the colon.
This research really sheds new light on why conditions like ICBPS and IBS often occur together. It's not just some random coincidence. There's actually a biological mechanism linking them.
Right. And it also helps explain why visceral pain, pain originating from our internal organs, can be so tricky to diagnose and treat, because it can be very diffuse and spread to seemingly unrelated areas.
It's like a domino effect.
Yes, exactly.
And perhaps most importantly, this research opens up exciting new avenues for developing new treatments for these conditions. Targeting this MRGPRX2 receptor, for example, could potentially provide relief for people suffering from both bladder and bowel problems.
I agree. And the fact that there are already drugs in development that target this receptor for other conditions is very encouraging. It could accelerate the development of new therapies for these visceral pain disorders.
This is really groundbreaking stuff, and it makes you wonder, could there be similar interorgan communication pathways at play in other parts of the body? Could this explain why some people experience seemingly unconnected symptoms in different organ systems?
That's a great question. And it's certainly an area ripe for future research.
It's mind-boggling to think about all the complex interactions happening within our bodies that we're just beginning to understand. Thank you so much for taking us on this deep dive into the fascinating world of interorgan communication.
It's been my pleasure.
It really does make you appreciate just how interconnected we truly are, and who knows what other surprising connections we'll uncover in the future. This is just the beginning of a very exciting journey of discovery.